Today we are continuing from where we left off last week. We started looking at the basic structure of amino acids. So we got to essential and non-essential amino acids. That's where we stopped. Today we'll continue and hope that by the end of today's lecture, we'll be done with the structure of proteins. And then we move on to look at enzymes. And from there, we can dive into the metabolism of proteins. So today we're starting with the classification of these amino acids, or let's say categorization of these amino acids, depending on the type of side chain that they have. So the last time we mentioned that all amino acids have the alpha carbon, on which you have the amine group, the carboxylic acid group, there's a hydrogen, and then there is a side chain. And it is a side chain that distinguishes between the various amino acids. Now, based on the side chain, we have amino acids that we can group as non-polar and aliphatic side chains. So aliphatic only means linear, so linear side chains. And those side chains are non-polar. So the first you see there is glycine, the smallest amino acid. The side chain is hydrogen. If you remember, we also mentioned that it is glycine that does not have a chiral center, because the side chain is hydrogen, which makes all the groups around it not different, because it is having two hydrogens on that alpha carbon. Now we continue from glycine, and to make the side chain, to see the more complex side chains from glycine, alanine, in place of that hydrogen, you are now seeing there is a CH3, a methyl group. That's another aliphatic group. It's non-polar. Proline, you see proline is having some intramolecular cyclic structure, because you see that the side chain, which is on the alpha carbon, forms a cyclic structure with an amine group there. But it's aliphatic. That side chain is not aromatic. You come to see when we are looking at aromatic side chains, you realize this is, so you don't confuse this as an aromatic side chain. This is a side chain that is forming an intramolecular cyclic structure with an amine group, and it's also non-polar. Valine, we see that also. So you see that it is just, addition of carbon atoms. If you see from glycine, there was one carbon added on that alpha carbon, and you have a methyl group CH3. Then when you go forward, proline, you see more carbons are added there. There's another CH2 and then a CH2 forming that cyclic structure there. Valine, you see also more carbons added on that alpha carbon. All of these are a non-polar side chain. Leucine, isoleucine, they are all aliphatic or non-polar side chains. Then you see there is a unique amino acid here, which is having sulfur in it. That's methionine. In fact, there are two amino acids that you will come across with sulfur in them. This is one of them, methionine. And you also will come, I think if you studied the synthesis of proteins in first year, how protein is synthesized, or if not yet, you come to realize later when you are doing a protein synthesis, how protein is synthesized from nucleic acid. You realize that at the beginning, the first amino acid that starts the process is methionine. So this is methionine. And you see here, there's a special bond that is existing in methionine referred to as a thioether group. You know, you refer to an ether. An ether group is a carbon, oxygen, carbon. So carbon bonded to oxygen and then oxygen bonded to carbon. So if you have in place of this sulfur oxygen, that is an ether group. So again, if you did some organic chemistry, you would have seen such a group, ether, E-T-H-E-R, an ether group. So when you have a sulfur in place of the oxygen, then you refer to it as a thioether. In fact, when you're doing biochemistry and you see, or even chemistry, and you see something like thio or thia, T-H-I-A, it's referring to sulfur. You quickly know that in that compound, there is sulfur in it. Now, one thing about these side chains, these hydrophobic side chains, is that they contribute to stability of proteins because they are hydrophobic. Hydrophobic will mean that they are going to contribute to insolubility of amino acids in aqueous systems because they are hydrophobic. Right, so they will control that kind of solubility of proteins. I mean, proteins generally they are sort of if you have fragments, peptides, you usually prepare peptide solutions in water, but in large molecules of proteins like that, these non-polar side chains contribute to regulation of solubility of protein in the aqueous system because they are hydrophobic. Right. Then we move to the amino acids with aromatic side chains. So you see clearly the aromatic side chains, they are ring structures on themselves. They are not forming a cyclic structure with, say, the amine group or carboxylic acid group, as we saw with proline. So that's a distinction in between, if you say the cyclic structure that the proline is forming is not an aromatic side chain. You can see it clearly here. That's it's a ring structure there in the side chain. Just the side chain, not with an intramolecular cyclic structure with the whole amino acid molecule. Now, there are three of these amino acids having aromatic side chains: phenylalanine, tyrosine, and tryptophan. Now, when you go to you see, I mean, so these two, you can easily remember them by, say, if you take, for instance, phenylalanine. You see why is it called phenylalanine? You remember alanine is CH3. So just one hydrogen is gone, and then the addition of a phenyl group, that is a six-sided carbon ring, becomes a phenylalanine. And just by addition of a hydroxyl group to that, you have tyrosine. So you can easily remember this structure, phenylalanine and tyrosine. You can even say tyrosine is hydroxylated phenylalanine. That way you can quickly remember. Tryptophan is having 2 rings. Have a you have a 5-sided ring. Inside that 5-sided ring, you have some nitrogen with hydrogen on it, and then a tryptophan there, and then a 6-sided ring fused onto that ring. Later, as you continue to do biochemistry, you come to see some of these amino acids. You find them in the active center of enzymes, where particularly reactions that are catalyzed by these enzymes occur, and they are involved in some processes of proton transfer and things like that. Now, one other thing, practically, when you go to the lab and you do some protein extraction, you do spectrophotometric analysis of your extract to measure the purity of the protein. Now, the wavelength at which you measure your protein solution is 280 nanometers. Why 280 nanometers? Now, 280 nanometers because in the protein structure, you find these amino acids, particularly tryptophan, which is having 2 rings, which is responsible for the absorbance of monochromatic light at the wavelength of 280 nanometers. So you see tryptophan is giving a high peak there. Tyrosine will also give some peak, but very much lower than tryptophan. So the amino acids that are responsible for the absorbance of monochromatic light of proteins at that wavelength of 280 nanometers are these, tyrosine and tryptophan. To a minute extent, yes, phenylalanine also can, give some absorbance there as well, the same 280 nanometers. Even when you are doing a extraction of DNA, nucleic acids in the lab, you also do some spectrophotometric analysis to check the purity of the nucleic acid. And what you do is that you measure the absorbance of your extract at 280 nanometers, and you also measure absorbance, I think at about 260 nanometers or so, because it is at 260 nanometers that you have absorbance of nucleic acids, whilst you have absorbance of proteins at 280 nanometers. And you strike a ratio of the absorbances, because what happens is you have proteins and nucleic acids in complex, so that when you do the extract, you want to know that your extract, how much of that nucleic acid is it that you have extracted is bound to proteins. So like there's some protein content in what you have extracted. It is not only purely nucleic acid. So once you strike the ratio, you'll be able to see that what I have extracted, actually there is this amount of protein in it. Because if you strike a ratio of the absorbance and the absorbance of nucleic acid, the ratio compared to proteins is like 2:1. It means you have twice more of the nucleic acid than the proteins, which have also gotten extracted into your extract that you have. So this is a theoretical basis upon which you'll be doing some of these analysis in the lab. So when you go, remember this theory that the reason you are measuring your protein extract at 280 nanometers is because of the absorbance of light by these amino acids, the aromatic amino acids, the amino acids with these aromatic side chains, particularly tryptophan and tyrosine. Right. Then another thing you can keep in mind is that you see tyrosine is having a hydroxyl group. Now this hydroxyl group is capable of forming hydrogen bonds. Remember, we just saw aliphatic side chains, non-polar, which we said they were hydrophobic. This time around, tyrosine having a hydroxyl group can form hydrogen bonds, which means it will be hydrophilic. So it can form hydrogen bonds with water molecules. Right. Now, we move to another group of amino acids that have polar side chains. But these side chains are not charged. The fact that they are polar doesn't mean they have some charge on them, because sometimes you can easily confuse that to mean that because we are talking about these charges on the carboxylic acid group and the amine group also carrying a positive charge there. So you may be thinking that it is polar because it is also carrying a charge. It's not carrying a charge, but it's just because of the components in the side chain that is giving it that polarity. So for instance, serine. Serine is having a hydroxyl group in it. And that is what is responsible for its polarity. We see 309 is the same thing. It's also having a hydroxyl group there in it, also responsible for the polarity of that amino acid. Now remember I said there are two amino acids with sulfur in them, and we saw one which was methionine. You are now seeing a second one, which is cysteine. So cysteine is having a side chain, CH2SH, and that sulfur hydral group is responsible for the polarity of that amino acid. We see asparagine and glutamine also in this group, and that group, this amide group that they have, the C double bond O NH2, is responsible for the polarity that is in these two amino acids, asparagine and glutamine. And so the fact that they are polar, having these hydroxyl groups, sulfur hydride groups, and these amide groups should tell you that they can form some hydrogen bonds with water. And so that's what is going to make them hydrophilic. So not like the aliphatic non-polar side chains, which are hydrophobic. When you go further now, you see positively charged or basic side chains. Now we see examples here. Lysine. Lysine is having an extra amine group in it, which is carrying a positive charge. So that is what is responsible for the positive charge that the side chain carries. So if you pay attention, you realize that all this while we are just talking about the side chains, and the side chains are shaded. So that should tell you, like I said in the first lecture, that the way to learn the structure of the amino acids is to focus on the side chains, because anything else is the same in all the amino acids. So you just have to focus on the side chains. And you see lysine, you see more carbons there, just 4 carbons, and then an amine group is on it. And that amine group is carrying a positive charge. You come to arginine. So arginine is having what we call a guanidino group. You know, the guanidino group is that group that has you see you have a carbon bonded to nitrogen, bonded to carbon, and then there's a double bond N on it. That's that group. Actually, this, let's take it from here. The group that is NH bonded to carbon, bonded to NH2, and then on the carbon, there is a double bond NH. That group is what we refer to as the guanidino group. You can, after this, you can look at the structure of guanidine. You realize that the guanidine molecule is having a central carbon on which you have 3 NH2s. You will see that. So that's why it's being referred to as a guanidino group. Then, so we see here there are positive charges. For lysine and arginine, it's easier because you can see the positive charges there. But when you come to histidine, you see there is no positive charge. So the conversation here is not about the side chain being positively charged, but the side chain being referred to as basic. Now, basic because this side chain can accept hydrogen ions. So you see, you have some double bond between these 2 carbons. You also even have a double bond here between carbon and nitrogen. And what it means is that these double bonds can be removed by the acceptance of hydrogen ions. And so a molecule that is accepting hydrogen ions or protons, that molecule is a base. So this side chain behaves as a base. Now, another interesting thing about histidine is that it is one of those amino acids that you find also in the active center of enzymes. Sometimes you find it together with serine, and they have what you refer to as proton transfer between these 2 amino acids, and it's a form of activation of enzymes. So when there's a proton that is transferred, like maybe this hydrogen is transferred from the nitrogen, and it goes to form some hydrogen bonding first with the hydroxyl group of serine, and then later it is transferred onto another amino acid. Then it sort of activates the enzyme, like the active center of the enzyme is charged, and then there can be a reaction between there can be an interaction between the enzyme and the substrate upon which the enzyme is acting. So I mean, this is just to mention, it's one of the mechanisms by which enzymes act. I think I said after this we'll talk about enzymes, because particularly we are talking about metabolism. And so when we finish looking at the structure of proteins, we will then look at the specific function of proteins as enzymes, and then from there we move on to look at the various metabolic pathways, looking at the metabolism of the various biomolecules. This ring is referred to as an imidazole ring. An imidazole ring. The ring that you find, that structure that you find in histidine is an imidazole ring. Right. Then we also have amino acids with side chains that are negatively charged. And this is due to an extra carboxylic acid group in them. So you see aspartic acid is having a carboxylic acid group in it, glutamate or glutamic acid. So when sometimes you see aspartate, when you are reading, you also see something like aspartic acid. It is the same as aspartate. The same way you see glutamate, sometimes you see glutamic acid. It is the same thing that is being referred to. And this is and these two are having extra carboxylic acid groups in addition to the original carboxylic acid group that is on the alpha carbon. So you see that. So you can compare these to like you see glutamate, and you compare it to glutamine, and you see glutamine is having an extra NH2 there. So there's an extra amine group in glutamine. Asparagine, you compare it to aspartic acid, and you also see that there in asparagine you are having an extra amine group there. So you have an amide, which is responsible for the positive charge that that amino acid carries. Here it is carboxylic acid group and it's carrying a negative charge. So you can do that comparison. Compare asparagine to aspartic acid and compare glutamate to glutamine and see. All right. Now we can move on and look at what we refer to as zeta ionic forms of amino acids. Now, if you pay attention, you realize that so far all the amino acid structures that we were looking at, they were actually presented in their zeta ionic forms. And what does a zeta ionic form mean? That's an ion that is having both positive and negative charge on it. Meaning it's coming from a German word, zeta. Zeta means a hermaphrodite, something like that, something that's having both sexes. And here you have an amine group which is carrying a positive charge, and you have a carboxylic acid group which is carrying a negative charge. And so that we refer to as a zeta ionic form of amino acids. But you also have a non-ionic form, which sometimes we can also refer to it as a molecular form of the amino acids. So you have fully here, the carboxylic acid group is written COOH, and you see the amine group is written NH2. So there are no charges on this form of amino acid. Now, mostly you see amino acids written in these zeta ionic forms because when proteins dissolve in water in a solution, in aqueous solution, these amino acids actually exist in these zeta ionic forms. So they have these dipolar ions that are in solution. So that is why you see it being presented in this form, the Zwitter ionic form. Now, what that means is that the Zwitter ion of an amino acid can act as a proton donor and at the same time can act as a proton acceptor. So let's take the amine group, which is fully protonated. It is NH3 plus. That NH3 plus can be a proton donor. So what does it mean? It means that this NH3 plus can give out hydrogen ion. So you see the reaction where you have a Zwitter ion. It gives out a hydrogen ion, and so that NH3 plus now becomes NH2, and then you have the hydrogen ions that it gives, so plus H plus, the hydrogen ions that it gives out. So that shows that that Zwitter ion is acting as a proton donor. The proton is the hydrogen ion that is being given out. Now, the same way, the carboxylic acid group, the COO minus, can be a proton acceptor, which means that in an acidic medium, this carboxylic acid group will accept hydrogen ions and become COOH. And so then you have that carboxylic acid group becoming a non-ionic form. It is protonated. So you now have a COOH. So it's a proton acceptor. So it means the Zwitter ion can be acting as a proton donor, where the specific portion of the ion which is giving out the hydrogen ions is the amine group. That's protonated amine group. And acting as a proton acceptor, it is the carboxylic acid group that is accepting the proton or hydrogen ions to become COOH. Now, the Zwitter ion, or say amino acids, are then referred to as ampholites, which is to say these are amphoteric electrolytes. What does it mean, an ampholite or amphoteric electrolyte? So this is coming from the Greek prefix amphi. You remember the word amphibian. What's an amphibian? An amphibian is an animal that lives on land and in water, both land and water. So the same way you have the amino acid acting, as we saw, as a proton donor and a proton acceptor. And so it's being referred to as an amphoteric electrolyte. Now, the same way, if you have a fully protonated amino acid, which is to say that the amine group is NH3 plus, so it's fully protonated, and the carboxylic acid group is COOH, so it is also protonated. And so it's having a net charge of +1, because you can see that the NH3 is carrying a positive charge. The COOH is full, it's not carrying any charge. And so the net charge from this amine group, NH3 plus, now on the whole amino acid is now +1. So that is a fully protonated amino acid. Now, that fully protonated amino acid, we say it is diprotic. It is diprotic, means that it can give out 2 hydrogen ions. So here is it. First, you see that fully protonated amino acid gives out a hydrogen ion, and that hydrogen ion is being given out from the carboxylic acid group, and so that group becomes COO minus because it gives out a hydrogen ion there. Now the net charge on that amino acid is 0 because you can see the carboxylic acid group is carrying a negative charge. The amine group is carrying a positive charge. So these two charges cancel out and it becomes 0. Now, the amine group, which is carrying a positive charge, can also give out a second hydrogen ion. And when it gives out that second hydrogen ion, it now becomes neutral. So it's now NH2. In this case, the whole amino acid molecule will be carrying a charge, a net charge of -1. That charge will be coming from the carboxylic acid group. So we have seen clearly that, yes, a fully protonated amino acid, can yield 2 hydrogen ions in this manner. And because of that, we refer to it as a diprotic acid. Okay, now, this brings us to what we refer to as isoelectric point or isoelectric pH of an amino acid. So, or even if you can have an isoelectric point of a protein solution, and that will be the pH at which the protein solution will be having all amino acids that are in it being in their zwitter ionic forms, which means the net charge will be 0. Now, if we take the smallest amino acid, glycine, as an example, and that amino acid fully protonated, the reaction which we just looked at, that the first hydrogen ion, which is given out by the carboxylic acid group, is having a pK value. Now, a pK value is, it shows the ability of a molecule to give out hydrogen ions, the ability of it to allow for the dissociation of hydrogen ions from it. And the lower the value, the higher the ability of the molecule to give out hydrogen ions. And we see that here you have a pK1 of 2.34. Now, at that at a specific pH, where you have some hydroxyl equivalent of 0.5, you can have that process of giving out of hydrogen ion by the carboxylic acid group occurring. Now we see that at a certain pH, say 5.97, which is the isoelectric point, you have the amino acid existing as a zwitter ion, meaning that at that point, the net charge of the amino acid is 0, because the amine group is carrying a positive charge and the carboxylic acid group is also carrying a negative charge, and these two charges cancel out. This is one of the things also you may be doing in the lab, looking out for the isoelectric point of protein solution, the pH at which the net charge of the protein solution you have is having a net charge of 0. I mean, you say that theoretically what it means is at that particular point, you have all the amino acids in solution existing in their zeta ionic forms. And you have a pK, the second pK value there you see is much, much higher than the first, which is 9.6, which is showing the ability of that zeta ion to give out the second hydrogen ion, which means the second hydrogen ion is given out with more difficulty than the first one, because that one is coming from the amine group, and that's even in a basic medium. In any case, the carboxylic acid group is an acid. That's an acidic group, and so it should easily give out hydrogen ions. The amine group is a basic group, so of course it will be giving out hydrogen ion with more difficulty. And in the end, you now have a negatively charged amino acid. So this reaction is just a reaction which we saw earlier, depicting that a fully protonated amino acid is diprotic. It can give out 2 hydrogen ions in those 2 steps. Now that we have looked at amino acids, which are the basic units of proteins, we can continue now to start building a protein where we can have amino acids binding. Now, the bond that exists between amino acids in proteins we refer to as peptide bonds. In some cases, they are also referred to as amide bonds. How does that bond form? So we take 2 amino acids, and one of the amino acids is having a side chain being represented R1. The second amino acid is having a side chain referred to as R2, more or less written like R squared. And we see that the orientation of these 2 molecules is such that you have a carboxylic acid group, which is in close proximity with the amine group of the second amino acid. Intentionally, we are showing the groups that are interacting. So we have a hydroxyl group, which is red on the first amino acid, and we have a hydrogen, which is also red on the amine group of the second amino acid. So it is this hydroxyl group, the red hydroxyl group, and the red hydrogen that are interacting between them, which will result in the formation of a peptide bond. So what happens? Water is given out. This OH and H react, and water is given out. And when water is given out, you then have a bond which is forming between the carbon of the carboxylic acid group and the nitrogen of the amine group. So that bond is what you see below here. And you see the whole of the amide group is shaded yellow. So you see the amine group, the peptide bond forming there between the carbon and the nitrogen. Now, again, you see here you see that there is a hydroxyl group that is interacting with a hydrogen and water is given out. You can easily confuse that to think that the bond which is forming between this, like the reaction which is occurring between these two amino acids is an acid-base reaction. But it is not. It is not an acid-base reaction. This reaction is a condensation reaction. In any case, you have a carboxylic acid group. So it's an acid. So if it should be reacting in an acid-base reaction, it should be giving out, or it is only its hydrogen that should be reacting, so that in the end you have a COO minus, as we saw before, that carboxylic acid group gives out protons and becomes COO minus. But this time around, it is this hydroxyl group which is reacting with that hydrogen. Now, the NH2 is also not an acid. An amine group is actually basic. So we can't say this is an acid-base reaction. It's a condensation reaction. Now, this type of reaction, you will see it occurring throughout. You see it over and over in different biomolecules, in carbohydrates, just like you have seen here. You see it again and again. So once you get it once like this, understood, you see that it will carry you through. And condensation reaction, sometimes also, being referred to as dehydration, because there's a removal of water. So you say dehydration. Condensation, like meteorologists will say condensation, like rainfall, formation of water. That's what it is. Now, this is a formation of a peptide bond between two amino acids. That will give you what? This molecule that you have here is a dipeptide. So that's a peptide containing two amino acids. So the continuation of this, so that if this reaction continues on and on, where you have this carboxylic acid group at the end here, reacting with another amine group, that is, if this carboxylic acid group is a COOH, and the same reaction occurs with another amino acid with the amine group, then it means the chain continues. And the chain can continue also on the left side when you have another amino acid reacting, the carboxylic acid group reacting with the hydrogen on the amine group. And the chain will continue to grow. So you have, from a dipeptide, you can have a tripeptide. That's having three amino acids in it. You can have a tetrapeptide. You can have a pentapeptide, hexapeptide, heptapeptide, and so on. And usually you have what we refer to as oligopeptides. So what we refer to as a dipeptide, all the way up to like peptides with 20 amino acids in them, are grouped as oligopeptides. And if you have a protein chain which is having more than, say, 20 amino acids in it, that is being referred to as a polypeptide. So when you read biochemistry, biology, and you see peptide, polypeptide, there are other ways of saying proteins. Usually, if you say polypeptide, you are referring to protein. Oligopeptide is a protein. Any peptide is a protein. So if you hear peptide, you are still saying protein. Okay. Now, another thing that you should pay attention to is that, you see, when these two amino acids bonded, forming the amine group, you see the end on the right-hand side is a carboxylic acid group. And the end on the left-hand side is an amine group. And I said the chain can continue to grow when more and more amino acids bind. What it means is that on the right-hand side, if more amino acids come to bind and the chain grows to the right-hand side, always the end of the peptide on the right-hand side is going to be a carboxylic acid group. As you see here, the end here is a carboxylic acid group. Then this carboxylic acid group, it will bind with an amine group. And so that the amino acid which will bind to it will still be ending with a carboxylic acid group on the right-hand side. The same thing will happen on the left-hand side, so that a carboxylic acid group of an amino acid will come to bind on the amine group, which means the end of that amine group will still be an amine group. So if you take a peptide chain, always there's going to be one side which is ending with an amine group, and the other side will be ending with a carboxylic acid group. Now, the side that ends with the carboxylic acid group, when you read, is being referred to as C terminus. It only means that the carboxylic acid end, C terminus, where the chain terminates, the end. So carboxylic acid end, C terminus. You also see N terminus, which means the amine group end. So these two terms, you pay attention. Sometimes they just draw some, it's not necessarily even, they will draw this whole structure, because for you to be having all the atoms showing, carbon, oxygen, nitrogen, all of them, it can make things clumsy. So sometimes you just see maybe a line is drawn, and then on one side they write N terminus, and then the other side they write C terminus. It's only telling you that this side, the side where it's written C terminus, that is the end where you have the carboxylic acid group. And where N terminus is written, that side ends with an amine group. Right. Now, from that formation of a peptide bond, we can move on to more complexity, looking at the different levels of protein structure. Now, protein structure is referred to in 4 different levels. The first is primary structure. Now, the primary structure of protein only refers to the sequence of amino acids that are in a protein chain or in a peptide chain. So, for instance, when you are referring to the structure of a peptide, and you are saying that, so what you see on the screen, from the top, you see lysine is bonded to another lysine, bonded to glycine, which is bonded to another glycine, and that is bonded to leucine, and that is bonded to valine, and valine is bonded to alanine, and alanine is bonded to histidine, When you are talking about that sequence, one amino acid after the other, one amino acid after the other, what you are talking about is the primary structure of the protein. Just the sequence of the amino acid, the sequence of the amino acids in the peptide chain. So that's a primary structure of the protein. Now you can have what you refer to as, somebody says you cannot hear me. Really? Well, sometimes it's possible it is your network, because I can see my network is very full. Everything is moving. But two people say they can't hear me. Anyway, don't worry. Whatever it is, after the lecture, the video will be shared, so you can watch it even multiple times until you get it. All right. So we see that apart from the primary structure, we also have what we refer to as the secondary structure. We will look at the secondary structure more in detail. We'll see two of them. And we'll also look at the tertiary structure, which is, say if you take a protein and it's having two or more secondary structure in it, that protein is having a tertiary structure. But we'll see, say, the bonds, the structure the bonds that are involved in the tertiary structure of protein. And we'll also look at the quaternary structure, where you are having two or more tertiary structures of proteins. Usually you have like protein subunits that are coming together to act as one molecule. So let's continue. As for the primary structure, is well understood. It's about a sequence of amino acids that are present in a peptide chain. So let's move to secondary structure. Now you can have two kinds, generally two types of secondary structures of proteins. The first one is what we refer to as alpha helix. Alpha helix. And the alpha helix is depicted on the right-hand side of your screen, where you see a right-handed twining of the protein chain. Now, if you want to be specific, in this structure, after every 4 amino acids, that is, if you take, say, amino acid, you number them, amino acid 1, then you number 2, 3, 4. The 4th amino acid, its carboxylic acid group that is having a C double bond O on it can form a hydrogen bond with a hydrogen on the amine group of the first amino acid. Now, this is what you see being depicted here, that you have this, it's like a ribbon. And in that ribbon, you see the hydrogen bonds being shown, that you see that there is a hydrogen on the amine group of one amino acid, and that hydrogen is forming, you see these dotted lines which refer to the hydrogen bond with the oxygen of the C double bond O, that is the carboxylic acid group of the 4th amino acid there. So you see these dotted lines are referring to those hydrogen bonds. So it forms some kind of a stack. It's a stacking. Sort of this ribbon, like a a staircase, a ribbon-like staircase. And you see these hydrogen bonds which are holding the amino acids together. That is the alpha helix structure of a protein. So, I mean, proteins, even though we refer to them like you see the primary structure, that this amino acid is bonded to this and is bonded to this. It's not like when you take a biological system like your body, the proteins are like rods inside you. No, proteins are folded. They are in globular forms. And this is what we refer to as the secondary structure of the protein, having hydrogen bond. So here you see what we were talking about earlier, N-terminus. And at the top, you see C-terminus. So if you didn't know what this meant, from today, when you see it, you know what it means. N-terminus, C-terminus. And you can also keep in mind which bond is involved in the secondary structure, the alpha helix of proteins. That bond is what? Hydrogen bond. We'll see another secondary structure. You see there too is a hydrogen bond. Now the second secondary structure we are looking at is beta sheets. This time around, you have a peptide or a protein which is folded in such a way that the peptide chain is sort of having some parallel and anti-parallel sides. So you see so this is N-terminus, and you see the protein chain goes all the way. Then it folds and comes down, anti-parallel, and then is going up again, parallel, and you see the C-terminus. Now here too, you see that hydrogen bonds are being formed between the oxygen of the carboxylic acid group and the hydrogen that is on the alpha carbon. You see these hydrogen bonds along the line that way. And this structure actually forms like a zigzag structure. What is being depicted here is like a zigzag structure. That is beta sheets. All right, so we have talked about two types of secondary structure, the alpha helix and the beta sheets. These two, the bond that is involved in their structures is the hydrogen bond. Now, the tertiary structure. I said a tertiary structure of a protein is a protein that is having two or more secondary structures in it. And this is being depicted here, such that if you take, say, the sides that are, ribbon-like, these sides are depicting, say, alpha helix, the alpha helix which we saw. And we even see that we are seeing protein strand that is folded like anti-parallel to each other, and we are seeing some hydrogen bonds between them. This is to depict the beta sheets that we saw. So we are seeing alpha helix, beta sheets, so secondary structures. And the alpha helix, we have even seen about 4 of them over here. and we are seeing one of the beta sheets. So you have secondary structures coming together to give you a tertiary structure. Now, what are the bonds that are involved in the tertiary structure of proteins? Now, the first we are looking at over here is referred to as a salt bridge. A salt bridge. And a salt bridge is formed between the NH3 plus, that's the amine group, and the COO minus, the carboxylic acid group. It's more or less like ionic bonding, but it's you know this coming from zeta ions, so that's why it's being referred to as a salt bridge. It's not a real salt that is being formed. Like if you have you know, when an acid reacts with a base, it forms a salt. So that is why, so for instance, if you have what? Hydrochloric acid reacting with sodium hydroxide, you have the salt sodium chloride plus water. So that is why this is being referred to as a salt bridge, because it's an interaction between, say, an acidic group, the COO minus, and an NH3 plus, more or less like some ionic group that is having some interaction between them, and is being referred to as a salt bridge. You can see this as a bonding between two secondary structures. So this is one alpha helix, and this is another alpha helix, and you are having that bonding between them. We see also hydrogen bonding. So here, this is the CH2OH, which you are seeing here, is depicting the side chain in which amino acid? In serine. We saw in serine, the side chain was CH2OH. So we see another CH2OH. So two serines can interact, and between them will be hydrogen bonding. So that's the hydrogen from one of the hydroxyl groups and the oxygen from the second hydroxyl group. Between them, you can have such hydrogen bonding forming. We see also hydrogen bonding forming between the carboxylic acid group, that COO oxygen, the C double bond O, that oxygen there is having a hydrogen bond between it and the hydrogen of an amine group of another amino acid. So this is also depicting another hydrogen bonding. Then we see a side which is depicting the beta sheets with hydrogen bonds between the antiparallel strands. We also see what is referred to as disulfide bonds. Now, where can disulfide bonds form? Obviously, it is going to be between amino acids with sulfur in them. And what are those amino acids? We saw two of them, methionine and cysteine. So between the sulfur in these amino acids, you can have such interaction, particularly where cysteine, which is ending with a sulfur hydral group. So that hydrogen which is on the sulfur would be given out, and then there will be a bond which is formed between the sulfur of these amino acids, the cysteine, and you have what is referred to as disulfide bonds. You can have what is referred to as hydrophobic interaction. Remember, we saw some of the side chains being hydrophobic, non-polar. And one of them is the methyl group. So like you saw the methyl group in an amino acid like alanine. So if you have CH3, CH3 close to each other, the interaction that is going to be between them is going to be hydrophobic. The same way, if you are having, say, phenylalanine, two of them close to each other, these groups are also hydrophobic. The interaction that is going to be between them is going to be a hydrophobic interaction. And at the top, you are seeing a depiction of hydrogen bonding with water molecules. So that is to say that if you have this whole molecule, this whole protein in an aqueous solution, of course, there will be amino acids in there which are hydrophilic, like this is depicting tyrosine, the ring with an OH on it can be having some hydrogen bonding interaction with water molecules. The same way, CH2OH is depicting the side chain in serine. That too can be having some hydrogen bonding interaction with water molecules. So here you have different bonds that are present in the tertiary structure of proteins being illustrated. And what are those bonds? Salt bridge, hydrogen bond, disulfide bond, and hydrophobic interactions. So 4 of them: salt bridge, hydrogen bond, disulfide bonds, and hydrophobic interactions as the bonds that you find in the tertiary structure of proteins. Now the quaternary structure of proteins. So here too, it means that you are having a protein that is having 2 or more tertiary structures. And usually these are subunits, like, you know, a protein which is complete protein on its own, which is binding to another complete protein, and they form a complex and they are acting as one. An example, major example that you see that we talk about is hemoglobin. Hemoglobin. So hemoglobin is made up of 4 subunits. So this is what you see here, hemoglobin made up of 4 subunits. And these 4 subunits are shown as you are having some alpha subunits, beta subunits being shown in different colors like violet and green. And you have these 4 subunits coming together to give you the structure of that protein, the quaternary structure, acting as one. And between these subunits, it is the bonds that you saw in the tertiary structure that are going to be existing here. There will be hydrogen bonds, there will be disulfide bonds, there will be hydrophobic interactions, and which other bond haven't we talked about? There will also be salt bridges over there. All those bonds will be present in the quaternary structure as well. On the slide is just showing you how from a primary structure where you have a sequence of amino acids in a peptide chain. From there, forming a secondary structure, which can be an alpha helix. Of course, you can also have beta sheets. And from there, you have a tertiary structure. And from the tertiary structure, you have a quaternary structure. So these are the 4 levels of the organization of proteins: primary structure, secondary structure, tertiary structure, and the quaternary structure. Right. Now, let's talk a bit about what we refer to as denaturing. A lot of times you may confuse denaturing to mean that there is a protein which is broken down into amino acids. But no, that's not what denaturing is about. Denaturing is not about breaking a protein into its constituent amino acids. No. Because denaturing does not involve the breakdown of, say, peptide bonds. The peptide bonds exist between amino acids. But denaturing is about breaking, you see the bonds that we talk about that form the secondary structure and the tertiary structure, which are also the same bonds which you find in the quaternary structure. So for instance, if you take, say, the hemoglobin that we talked about, the hemoglobin is having a particular structure, and that structure is what makes it functional. If that structure is broken down, the molecule will become dysfunctional. Now, that structure can be broken down when hydrogen bonds between the tertiary structures, the secondary structures are broken down. When the salt bridges are broken, when the disulfide bonds are broken, when the hydrophobic interactions are broken. And what are the things that can break down these bonds? Things like high temperature, can be, say, low pH, high acidic medium can cause denaturing, or high basic, high pH can cause denaturing. There are some heavy metal salts that can also cause denaturing, or just agitation. So for instance, here we are showing an egg. The egg is having, in it, you have the yolk that's having some protein in it that is, holding it in that form. But if you beat it, it's scattered. So you have broken down. So agitation can break down some of these weak bonds as well. And so you see here being shown like an active protein is having some of these bonds being shown, like hydrogen bonding, disulfide bridges between them, like shown in this orange, yellow, green colors, to show that the bonds that are between the various parts of the protein. And then a denatured protein is having those bonds broken off. So that is denaturing. So it's not about breaking down peptide bonds between amino acids. No. The breakdown of peptide bonds in amino acids, already that is going to be in metabolism of the protein, breaking down the protein into amino acids, which will then come together now to synthesize the protein that the body needs. All right. Now from here, we can briefly talk about the fact that, you see, so far, as we've talked about proteins, I want to end with the fact that, you see, proteins can have different functions in the body. And I said, because we are coming to talk about metabolism of different biomolecules, we'll focus on this first function, which is proteins acting as biological catalysts, as enzymes, to accelerate the rate of chemical reactions. You can also have proteins being like structural proteins, forming body tissues, like you have maybe keratin in the hair, the nails, collagen, maybe in the cartilage, that is, giving some cushioning to the joints. Proteins can serve as, some transporters in the body. So, for instance, you have some cell membrane proteins. So you have some transmembrane proteins that are involved in the transport of substances across the cell membrane, either from the extracellular space into the cytoplasm or from the cytoplasm to the extracellular space. So transport proteins. Or you can talk about transport specifically, say oxygen transport, which you'll be referring to hemoglobin. Not only hemoglobin, there's also myoglobin. So hemoglobin will be involved in the transport of oxygen from the lungs to the tissues. And when it gets to the tissues, you have myoglobin, which will be serving as an intermediary, receiving the molecules from hemoglobin one after the other and transferring it to the tissues. And carbon dioxide as a byproduct partly is carried by hemoglobin to the lungs to be exhaled out of the body. Then we have proteins that can also be acting as neurotransmitters. So this for signal transduction in the nervous system. You have some of these proteins that will also be acting in that manner. Or you refer to what we say immunoglobulins. What are immunoglobulins really is, they are white blood cells. These are involved in immunologic defense, the immune system. We can also have proteins. Say we have some peptide hormones. So proteins as hormones, which are involved in the regulation of biochemical processes. Some proteins form complexes with carbohydrates, what we refer to as glycoproteins. And these act as receptors, say on the surface of, say, the cell membrane for recognition of some specific signals. So, Salom, I'll respond to you when I finish with this. I'll respond, so don't worry. Then you have, you can even have something like lipoproteins. So you can have nucleoproteins, so complexes, which will be acting in different ways. So you see, just protein alone can give you different fields that people spend their whole lives only in a specific field. Like, say, if you are talking about enzymes, that can give you a field on its own referred to as enzymology. And people study enzymology all the way to advanced level, have PhD, become professors, just enzymology. Or even if you talk about, say, okay, neurotransmitters, for instance, that's neurobiology or neuroscience. That's a whole field on its own. Or you take immunologic defense, you have a different field. Immunology, you have proteins acting as hormones. That one too is in a different field. Endocrinology. So you see, just proteins alone can be broken down into specific fields, and people can spend their whole lives, become experts in a specific field of these things. So you see how very interesting biochemistry can be. So you should like it. You should love it. It's about, a lot of it is about what is occurring inside you. So it's something which you should love. All right. So now I see Solomon Adigbo says, sir, please. So is it inadequate protein in the body that causes hair on the head to fade, or it's a disorder? Okay, so I think what you are referring to is like baldness. Baldness. So baldness somehow is sort of genetic, because in some families you see baldness, in others you don't have such. But some of the baldness too can be as a result of exposure to some medication, some chemical. There are some people, maybe because they use some sort of hair products, some cosmetics, some drugs, can result into baldness. Some maybe not better care have been taken on the hair and has been exposed to so much of, some adverse conditions. So different things can cause baldness. Autry Daniel says, sir, it's not easy. So what is it that is not easy? Samuel Ansah says, does it mean that we must always eat foods containing protein? Of course, you must eat balanced diet. Eat food not only containing protein, but other nutrients as well must be there. So not only protein. All right. So apart from this, you can continue writing. I'll be responding, and then if anybody wants to say anything, the person can raise the hand, and then I will enable the microphone of the person. I'll call your name, and you speak. You disable. You will unmute yourself, and then you speak. Then I can respond. So Hilda Bassa says, "Sir, that's protein helps in weight loss." So it's not only protein that will help in weight loss, but of course, sure, because protein is involved in tissue repair. So if there is weight loss, of course, protein plays an important role in tissue repair, so can help in terms of weight loss. Okay, anything else? So the floor is open. You can write in the chats. You can raise your hand, and I will enable your mic. I'll call your name, and then you You say what you want to say, whether it's a question or it's a contribution you want to make, anything. You say it, and then we can respond to it. Felix Cujo says, so, sir, what about cancer? When people lose their hair, I think you want to say, is it about inadequate protein also cause of the lost hair? No, actually, it's not. It's not about in cancer. It's not about inadequate protein. A lot of times, hair loss in cancer occurs by even exposure to some of the medications, like in chemotherapy. It causes some of these hair